Welcome to the lecture over gene expression and mutations. I'm going to start off with a recall. We know that chromosomes are divided into segments called genes. These genes are directions or instructions for building all of the proteins needed by an organism. And so when they build those proteins and the proteins are produced, then they are going to express the gene. So what is gene expression? It's when the product of a gene or a specific protein is being actively produced by a cell. Not all genes are expressed or active at the same time. Some genes are rarely expressed, like adrenaline. We only express the protein adrenaline to be created for when we are doing our fight or flight response. Some genes are constantly expressed, like hair growth, blood pressure, things like that are constantly expressed. And then some genes are expressed for a time, but then turned off. Those are cyclical genes, like estrogen. The estrogen would only be produced in high levels and expressed during a female's menstrual cycle. And so during that time of the month, they will be expressed during that time, and then they will be turned off for the rest of the month. And so not all genes are going to be expressed at the same time, or they might be expressed in some cells but not other cells. So why aren't they all expressed or active at the same time? They don't want to produce too many molecules that the cell doesn't need. This would be a waste of energy and materials. So if, for example, red blood cells need hemoglobin, a protein, to bind to oxygen to carry the oxygen in the bloodstream, but the other cells don't need the protein hemoglobin made, then the cells that are not red blood cells are not going to have that gene expressed or active in those cells because they don't need to create the hemoglobin protein. Another example is they're not going to express genes to create hydrochloric acid in our eyes, for example, like we do in our stomach. It would be very painful if we were expressing hydrochloric acid in our eyes and instead of our stomach. And so we only express certain genes in certain cells at the same time. But that doesn't mean that the instruction for those genes are not in every cell. They are. The instructions are in every single one of the cells, but it means that they're not always actively expressed. So why is it important that we regulate this gene expression? We know that different kinds of cells require different kinds of molecules to function. An example is a heart cell in your body has the same DNA as a skin cell, but they look completely different. And that's because different genes are expressed in the heart than the genes that are expressed in the skin, because they have different functions in our body. Gene expression occurs because only certain pieces of DNA or the instructions are sent to the ribosome in each cell. And so if it's something that's not going to be expressed, you're not going to end up seeing the mRNA produced to send to the ribosome to produce the protein for those genes, because you don't need to create those proteins. You're only going to see the proteins created on the genes that need to be expressed in that cell. So the different proteins made is going to end up being the different genes expressed. So we know that the genes expressed have to have proteins made to express those genes. And this is just another reminder about cell differentiation. You know that all your cells have specific functions and they're very specialized, and so that is why the different cells are going to have different genes expressed in the different cells, because those cells have different functions, they're very specialized for that area, and so they're going to need different molecules or different genes expressed in order to function properly. We're going to review mutations as well, because with gene expression comes mutations at times. There are a different variety of mutations, and we're going to look at those again. We know that a mutation is a change in the DNA. These can occur when DNA is replicated or during transcription or translation. And there are several different types of mutations that we're going to look at. So if you see a change in DNA, then the DNA has been mutated. And you can also mutate the mRNA and tRNA and stuff like that as well. So there's different types of mutations. The first one are gene mutations. And these ones occur in DNA sequences, in the bases specifically. And so we talked about point mutations like substitution in our last unit. And we also talked about deletion or insertion, which is called frame shift mutations. We're going to talk about why it's called frame shift in just a second. And then there's also chromosomal mutations, and these occur at a macro level, at a large level of the chromosomes. That's things like getting an extra chromosome in trisomy 21, or not having enough chromosomes. That is a chromosomal mutation. We're not going to focus on that in this unit right now. We're just going to look at gene mutations, but I do want to make you aware that there's different types of mutations out there. So we're going to first focus on gene mutations, which is changes in the bases in DNA. So the first type was the point mutation, and this is where the mutation is happening in one location, but it's not changing the entire sequence. So it changes just one base pair in the DNA, and it affects only one amino acid because it's being switched. So here's an example. We have in our DNA here TACGCA, but we see a mutation where it says TACGTA now. So that C has now been substituted for a T. Now when we go through protein synthesis, we're now going to see that that change is going to cause a change in the amino acid sequence in the protein. So GCA originally would go to CGU. Using that codon chart, that originally would give you the amino acid arginine. But since we've changed it now, the mRNA for GTA is going to be CAU, which now codes for histidine. And so one point mutation can change one amino acid. It could also cause no change to the amino acid if the mRNA sequence is the same amino acid as the original. If you remember from our notes before, that there are very many different amino acids that code for multiple. So you see CCU, CCC, CCA, and CCG all code for proline. So if the mutation ended up causing a change in this last base here of this codon, you wouldn't see a change in the amino acid sequence because all of these create proline. But if it physically changes the amino acid, then you're going to see a change in the protein. So once again, substitution is where one base or one nucleotide is changed for another. And so you see just this one base is changing, and the amino acid might change depending on how the mutation was. So here's another example. You ended up having mutation from the original sequence. It can cause a change in the end result of the sequence. A frame shift mutation can affect all amino acids after it because it's physically shifting the frame. If you remember, we read these sequences in groups of 3 when we're doing our codons. And so if we add a base in or remove a base out, it's going to change the entire rest of the sequence because it's going to shift it either backwards or one forwards. And so the two types of frame shift mutations are called insertion or deletion. Insertion where you insert a base, or deletion where you remove a base. And so here is an example of that. You see this TAC has been replaced with TAT, so there is an insertion of a T, because the C is still here. There's an insertion of the T in the sequence. But that also shifted the entire rest of the sequence. C is now the first in this codon, G is now the second, and C is the third. It's all been shifted down, so you're seeing a change. Now because they have all been shifted down, the mRNA sequence is going to be different, and the amino acids created for the entire sequence is also going to be different depending on how it shifted. And so this is called a frame shift mutation. So once again, it shifts the reading frame of genetic message. It may change every amino acid that follows the point of the mutation, and this can also make the protein unable to function because you have all of the incorrect amino acids after that mutation. So whether it's a deletion or an insertion, the base being removed or inserted, once again, is going to cause a whole frame shift mutation. So what causes gene mutations? We know that they can occur spontaneously in the cell, let's say during DNA replication or when they are making the mRNA, for example. We also know the environmental agents like UV light, radiation, carcinogens, cancer-causing agents, chemicals, cigarette smokes, viruses, HEB, right? So you will want to go back to your last unit if you don't remember all of these causes of mutations to review and remind why we have those mutations.